what we're going to do now is continue a little bit with some more extensive examples, and then I'm going to end with a few overview slides of all the kind of things that are possible. How am I doing in time? Doing great. So once you are using the platform, you can also appreciate that not everything is about gene expression. You can also have questions that are based on just the metadata, just the grouping variables that are in the platform. And for that we have this annotation tab. For example, what you can do is you can use this group annotation plotter. What that allows you to do is you just make a selection of the metadata variables that are already implemented in the database. You can drag them from one side to the other and then, for example, show them in a Sankey plot, such that you can immediately see how particular associations, in this case of subgrouping, different algorithms that have been used to subgroup these patients, how these patients are actually going about from one group to another. By just changing the order, you change the order of this as well. You can add or remove groups if you want to. And there are also different ways in which this plot can be done. So Sankey plot, which is one that we like now the most because it looks most beautiful, but you also have other kind of visualizations that can be used. And also there, if you click on any of these guys, it will actually zoom in, zoom out, such that you can see these associations and play around with maybe a group intersection that you want to explore deeper by making your own subtract for it. Other kind of visualizations that you can use is the annotation plotter. So annotation plotter will use the, again, track information, group, metadata values, where you can, in the order of your different tracks, you can just segregate your cohort deeper and deeper, which are these visualizations that you sometimes see in papers as well, if a cohort is being described. And also here you can just drag things from one to the other and then make this visualization as you see fit for your publication or your presentation. A third way in which you can work with the annotation, which is something that becomes a lot more powerful if you have particular questions, is you can use the cohort overview itself. So this presents itself as this pie charts. And these pie charts are also interactive in the sense that if you click on that, it essentially will just zoom in to that particular group, update the table here as well. But you can also use the table itself to make a particular subselection. So you want to exclude or include a particular set of patients. And by doing that, you basically are making a new sub-cohort out of your complete series of patients that you may want to have a deeper look at. So this you can then use as a basis of building a new track. So you can actually store that then as your own grouping variable and then later on do a differential expression analysis on it or whatever you find of interest. This also has some very powerful features that allows you to make subgroupings in it, but that may go a little bit too far for just an introduction, but just know that there are many different things that are possible also by using these tools. We already had a look at survival data in the beginning in the example with the Kaplan scanner. So we have a special domain, a special block in the R2 platform, which can be identified from the site where you can actually look at survival. And there you can also start doing these either Kaplan scanning on one gene, but also on all the genes in the genome. Or you can use other kinds of algorithms. So since I've already shown you the Kaplan scan, I'm here going to show you that you could also use an algorithm called Cox proportional hazard analysis, which is done a lot in biology as well. So what you can actually do is you can do this for a single gene, or you can do this for many genes at the same time. Here the example will be, we're using the complete data set. So we're back at colon cancer again with these 1000 samples. We're going to run a Cox proportional hazard analysis, including all of the genes, looking at, in this case, overall survival. And then after a minute or so, R2 will come up with the result, which is then a table that you can see at the bottom here, and a so-called forest plot. And what essentially happens, if those two things are linked, you can immediately then just work with p-value cutoffs, you can include or exclude genes, and you can change the sorting, and you, as you can see on the top image, everything keeps on being aligned with the selection that you're making, and in that way you can identify interesting relationships as well. If you find something of interest, you can click on it, and it goes now back into this Kaplan scan module, where you then can also see what it looked like from the Kaplan scan perspective. And in this case, it homed into a gene called NPR3, where you can see that there was quite a nice significant association with the cross-progression analysis. You can see it also works quite well in the Kaplan scan, where you have a very nice Bonferroni corrected p-value. So then you can also start to wonder, is this something that's only happening in this particular one data set? So is it really special there? So, or can I actually replicate this already? And for that, we have this survival analysis using multiple data sets. So what you can do over there is you can indicate by just selecting out of all of the data sets that we have survival data for. Here I zoomed into everything that mentioned colon. So colorectal cancer space, hopefully. And then I just selected all of them. And what I'm going to do now is I'm going to use this gene that I just discovered with the question, if I look at this particular gene, can I replicate what I've seen in my first data set, right? So is it something that happened more often, because that's something that can be used as a validation then immediately. And then just run the analysis, and then R2 will look for every data set what kind of survival data was in it. Sometimes it has only overall survival, other times it has relapse-free, or whatever you can think of. And it automatically tested everything, makes a new forest plot out of it, and here you can hopefully appreciate that since it did not span the odds ratio of the hazard ratio of 1, every time it finds a significant association in this case 22 different data sets. Meaning that this one does replicate, right? So this relationship between survival and this particular gene is not something special to the newness data set only. It actually happens in everything that we tested, which can be used as an argument that this is an interesting gene to have a deeper look at. There are many more options that you can actually do with the survival data, but I'll just leave it here for now. And if you are interested, have a look at our tutorial book, where actually more of these type of analysis are being shown, and also how you can actually use them. And yeah, sure.
So for the proportional hazard, how do you make sure that your assumptions of the test are tested? Like if you don't know what so not all data is fitted for a proportional hazard test?
Yeah, so here we made the assumption that since we're working in log space of the gene expression data, that's a sufficient parameter.
Data, is there a way to check if your assumptions are made with all these tests? Or
Not for the Cox proportional hazards. We haven't implemented that. It is something, of course, that can be implemented, because it's all of these analyses are actually in the back run in R sessions and these kind of things. So we actually can just extract these features out of it. Make that a column that can be used in conjunction. Yeah. So another type of analysis that you can perform in R2 is looking at heat maps, at gene sets. So just groups of genes that you may find of interest. For that we have the view a gene set option. There you select an intersection of genes that you may find of interest. So you're going to select a gene set. But you can also just add a number of genes manually if you want to. There's a box for that as well. We are again in this database of all the signatures that are already in there. And here I've tried to find an EMT signature. So from the hallmarks, again, I used epithelial to mesenchymal transition. And then once I've selected that, R2 will produce a heat map using only genes associated with EMT. We've already seen this one, of course, before, coming from a differential expression analysis. But here you can see that there's a quite nice signal for all the different genes showing kind of the same thing, which is pretty cool if you have a gene set. So normally what happens is that everything gets hierarchically clustered, but you can also tell R2 that you are interested in a particular intersection. I would like to have this seen as CMS because this is what we find interesting today. So you can just make that appear as a blocking variable where it essentially just makes all of the different groups look, put things together, and then only internally starts to cluster things, such that you can immediately see how they segregate. And then hopefully you can appreciate that the CMS4 group, as you can see over here, appears to have like higher expression in most of those genes. So the concept of seeing that particular pattern in a heat map is what we've also used to build our signature creation module. Because essentially what we do, if we build a heat map, we add this blue to red colored stripe underneath it. And this is what we call a signature score. And how the signature score is being calculated is essentially it's taking all of the genes that are mentioned in this particular signature. These are already in z-score space, meaning that everything has already kind of been normalized towards each other. So we're just going to have a look at, can we identify or can we make one signal to do like a majority vote for all of the genes and then represent this as one separate value. And if you do this, then you get the signature score. So it's the average of the z-scores basically. And then once you have that, you can use this information as well by using R2, by just saying that, hey, this score I find quite interesting. I want to use this as like a meta variable. I want to represent this as one new group, as one new tracking. And then in that sense, you can just store it in R2, and then it becomes a new grouping variable that you've added to your account. And that grouping variable can be used as if it is a gene by itself. So what we can then do is we can then use this EMT signal as a signature, which we're going to look at from that perspective, and then can again segregate this over the different subgroups that we had in our data set, the CMSs again, and again show that the EMT signature really has a higher value in this CMS4 value. If you get the concept of these signatures, then other questions that people started to have once this became an option is that, hey, we can also then just treat any gene signature as if it is a gene. So maybe we can do a whole scan out of that. So we can use the activity score, that basically now is our EMT signature, and then represent this against a database of all other kinds of signature scores that can be calculated on the fly. And this is basically what we do with the gene set versus gene set correlations. So we're going to here again identify, we started with this EMT as a signature, so this is going to be our X values. And as a Y value, we're going to use a whole database, in this case hallmark signatures from Broad, and see if we find an association between the signature scores. So these are now becoming activities instead of single gene expression values. And then again, you can see that there are quite interesting parameters in there. You can see what the correlation value is between the signature scores, how many genes are overlapping between the signatures, which is an important aspect if you start using signatures, because if you have 2 times the same number of the same genes in a signature, you will get 100% correlation. If the number of genes is very limited, but the p-value is very significant, then at least you're looking at different genes, so that is not going to be the reason why you find this correlation. And perhaps this helps you identify some interesting properties, some interesting pathways that can then be used to dig deeper. So here an example of this EMT signature versus here angiogenesis as a signature. That could be something that is of interest to have a look at. These can also be immediately digged into deeper because we have this signature. We know that it is there. We immediately have a link that you can just visually represent this as a heat map and then identify a gene that may be of interest there, such that you can just completely take this as a round trip again to really go deeper into what kind of properties may underlie this association and whether that would be interesting to follow up in experiments. So other type of meta-analysis that you can do, so not just using the genes directly, but also scores that you can associate from it, is you can run principal component analysis immediately from within the platform. So also here, what you do is you identify the group that you would like to identify. It automatically runs a principal component analysis for you. You can color these elements just as with all the other analysis that we had thus far. And then in that way see what the relationship between the principal components are of this data set and how that may segregate with a variable that you find of interest. And in that way you can also explore and work with that data to identify subgroups of patients or use these parameters as other covariates in the next analysis that you may have. Something that actually makes use of principal components in the back, but is something that's a little bit more advanced and that we typically see, for example, in single cell analysis that are also available in the platform. You can also make use of algorithms like TISNE or UMAP, which we actually have embedded as tools in the platform. So you can actually run a TISNE or UMAP analysis from within the platform. And once these have been created, either by us or by a public resource or in private, and then they become private to your own account, you can also reuse them. Once these results have been created, R2 stores them, and then you can actually use this as a kind of like a browsing portal to go through all of these types of analysis. And I'm going to show you today this browsing part, which you can identify here by the thing that we call sample maps. So in sample maps you will find TISNEs, you will find UMAPs, but also publication coordinate systems that we get from publications, from authors that are providing us these data. And then people can use a publication, for example, as the basis of looking at the expression of a particular gene. So what does it look like? So essentially you have this whole list of records that you can have a look at. We of course are interested in this colon data set again, so we're going to use this bulk colon data set, run here an analysis. This is a UMAP analysis, and then it ends up on your screen as this black dots. So also here we can color this by a track. Again, we're using the CMS classification here. You can visually adapt these plots. R2 does not only run one UMAP analysis, but it actually runs a number of different parameter options for it. With TISNE, it does like 45 different ones. With UMAP, it also does around 15 to 20 different parameter options, meaning that you can essentially see what the difference is if you just tweak the parameters a little bit. And contrary to what you may expect, that can have quite a drastic effect. And you can also then use these colored groups to highlight particular subgroups. You can switch things on and off. You can zoom in, zoom out, pan on it. And in that way you can actually explore what these results may actually mean in terms of subgroups or whatever thing you find interesting. Next to just using these annotations, we can also say that we want to color them by the expression of a gene. And then in that sense, you can just use, again, things like Vimentin or something like that to see where the expression of a particular gene is in such a constellation. And this allows you to actually go quite deep into these analysis, identify something that may be of interest. You can use lassos or dbScan, which are like algorithms that can identify either subgroups or where you can by yourself identify groups of samples that you find of interest and that you can then again store as a track and then use for later analyses. So depending on what kind of data set you have, if there are multiple layers available for a resource, like gene expression, copy number, signature scores, all these kind of things, R2 also allows you to take that level of information on top of these UMAPs. And if you want to play around with that later on, things like DepMap have multiple layers of data, and then you can actually play around with that kind of feature if you find that interesting. So I already said 2 or 3 minutes ago, so we also have, next to these bulk data sets, we have single cell resources that are available in the platform. Here just an example of one from a colorectal cancer space. So also there you can use these different tools to represent the data in such a way that you can explore expression of genes, look at subgroups, but you can also use tools like the 2G view, where you can show the expression of a particular gene in these so-called pi by group plots, which essentially show you the expression of a gene. So what is the proportion of the expressed versus not expressed, and then what is the average expression in this group, so that you immediately can get like a quick overview of how the expression of a gene is segregating over the different groups. But we also have implementations of these like bubble plots, where you essentially can use a grouping variable to segregate your cohort versus a number of different genes, and in that way also represent this in different kind of ways to show or to convey the message that particular sets of genes are over a particular subgroup has high or low expression of your gene of interest. And then the last example from this sample map space is we also have quite a number of publications where we've asked the authors for their coordinate systems of their figures, and quite a number of them have agreed to actually send us these coordinates, so you essentially can then just read the paper, see that, hey, there's a nice visualization around this, I'm interested in my gene of interest, can I actually also see maybe what my gene of interest is doing on the embedding on the coordinate space of that author. So this can then also be used directly from within the platform. And on top of that, we also have added our own analysis on such a resource. One of them being that we can use this whole UMAP Tisney scanning principle to see how robust is the result that the authors were giving, because we use the grouping information that came from the authors. But we also have this patched visualization, where this is an integration of 20 different samples. Every sample on its own has also been analyzed and put together in one view, so that you can immediately see how things like tissue or expression of a gene actually looks at it from the perspective of a single sample analysis. And in addition, we typically also have all of the separate samples as entities on their own, so that you can also have a look at those. Just an example of these kind of things, and we have quite a number of those. So the last part that I'm going to show you on some features that we have in the platform is some snippets around things that typically are not just gene expression, which you also have quite a lot of analysis implemented in the platform. One being that you can also use methylation data. So we've got, I think, around 50,000 methylation profiles already preloaded in the public part of the R2 platform. So these are typically things that you don't really look at from a one probe or one reporter perspective, but where you actually want to take into account the fact of how is the methylation property before or after a particular CpG island. So for this we have heat map visualizations where the order of the genome is instructed, and where you can then just have a look at what is the methylation status of this particular region, and that you can subgroup by diseases or by disease states, and in that way inspect and see the different patterns, which is also automatically linked back to the genome, so you can immediately see on average how things are affected. Using subsets, you can then just say, I want to look at only those two different groups. And of course you can visualize this by linking through to the analysis tools that I already showed you before. Multiple gene view can be abused to also visualize these types of data, and R2 can just constantly send data from one way to the other to make this visually possible for you. Another thing that we've been quite busy with, and where we have made a lot of developments, is looking into whole exome and whole genome data. So we're doing this for a number of precision medicine programs, also in the Netherlands as well as abroad. And this also allows you to make to have a more patient centralized view of a resource. So typically this zooms into one particular sample, one patient. In the example that I've been using thus far from the Nunes data set, so this is these 1000 colorectal cancers, there's also whole genome sequencing data available for it. So essentially this is also the easiest way to show you how that actually works. So for every single patient, we've run a separate analysis to reconstruct their genome, and then in that way make these like circle visualizations. These circle visualizations show you copy number states around here. The different chromosomes are in color code here. And then the links that you see, these are structural variants that have been identified in this. And essentially you can just click on any of those elements and then it zooms into this particular patient. Then you have a zoomed in view of the circle plot, but you also get all kind of panels with additional information on that particular patient. So you can see what metadata is available, what kind of structural variants are in there, which mutations have been identified, are there associations with known databases like ClinVar or Civic, so is this something that can be targeted? If it's multi-omics, like in this case it is, you can also attach the gene expression data to it, and in that way you can visually analyze this particular patient a bit deeper. So if you, for example, look into the gene expression space, then we actually look up for this particular patient in the gene expression dataset, which is their location, and then you can identify which gene is really highly over or underexpressed if we compare it to all the other 1,000 samples, right? What is special in this particular patient? And then you identify genes here that may be of interest. You can click on these guys, and then it goes back into this 2-gene view, where you can now see the segregation of the, in this case, the CMSs, and where this particular patient then is actually landing in terms of their gene expression. So you can really see that it's very highly expressed with respect to all the other ones. So this is kind of an analysis that you can also do for all kinds of genes. And there are many additional tools that we've implemented that are really specific for tumor board kind of perspectives, but which you can also explore if you are interested in it. Next to that, you can also look at it from, of course, the copy number perspective. So I already told you we have a genome browser in R2 which you can use. So for every gene that's identified in the genome, it also looks at, is there something happening around that particular region? And then you identify in this case that CDKN2A appears to be homozygously deleted in this particular patient, which you can see because the copy number state is really 0 with respect to the rest. And that way you can also integrate the information in different layers and explore this deeper. That was looking at it from a single sample perspective. We also have things like oncoplots implemented in the platform, where you essentially can use the whole cohort again. So now you can just have a look at different genes, all of our patients, and see how often a particular gene is being affected on either copy number, gene expression, fusion kind of relationships, and thereby also make these visualizations. And these visualizations can be instructed by using tracks or more than one track to sub-segregate everything. You can make layers in this and then create pictures that may actually show what you're trying to say with your figure. And on top of that, we also have a discovery module on this where it's going to use visual exact tests or chi-square tests to associate any of those genes that we identify over here against all of the clinical features that we have. So we essentially can constantly just brute force test every possible combination to identify, is there a particular gene that shows an interesting association based on all of the criteria that we've used here to build the oncoplot. And it kind of goes a bit far, but you can actually do nearly infinite kind of combination possibilities here to say I want to include only genes with this particular overexpression, only those copy number state, only those genes, and in that way you can really define a particular cohort and identify something that can be quite interesting. Here, one of the things that you actually may have expected. So in colorectal cancer, you have this concept of microsatellite instability, which typically happens if particular repair genes are broken, are mutated. And one thing that you immediately pick up is that if you have MSI status associates with MSH3, which is one of those repair genes that then is broken. And also here, because we can use colors on these visual Fisher exact tests, you can immediately appreciate that this appears to happen only in the yellowish kind of group, which is the CMS1 that has as a feature microsatellite instability. So you actually can just use all of the information in one go in a single visualization. My next slide. Interesting. So other ways in which you can look at the cohort is looking at it from mutation space, so you can actually identify particular mutations. You also make collections out of this, like in colorectal cancer, like APC is, for example, a thing that we would like to have a look at. So you can identify APC mutated cases, store this as a track again, and then use gene expression or other types of information to have a look at that, because R2 can also combine these different resources on top of each other. You can also have a look at this from the copy number space in overviews, and these overviews can also be quite interesting. So here again I've used these 1000 colorectal cancer samples. The visualization here is that if you see that it's gray, it means it has 2 copies. If it is blue, it means that it has lost 1, and if it's dark blue, 2 copies of the gene, and if it becomes more and more red, it has more copies, right? So gains and amplifications. And that we can now also segregate by using tracks. So essentially we can say, hey, what happens if I use CMS status as here? And then you can immediately see that if you have a CMS1 type tumor, that typically has a less busy landscape, because there the mutational process is using this microsatellite instability than, for example, others. And in that way you can also use any track, any feature that you want to explore these data deeper. Once you find something of interest, you actually can go into the genome browser, and there you even have more possibilities to make all kinds of adaptations to that particular intersection as well. Still, on top of this personalized medicine, like whole genome sequencing view, you can also use other tools that we have implemented in the platform. Here there's an example of MAF tools, where you can look for somatic interactions, so which genes appear to be co-mutated and which are never mutated if one is mutated. That can also help you identify interesting patterns in the data. And also here you can visualize this in different ways. Last part that I would like to show you before having a couple of housekeeping slides and going for the tutorial part. Chip sequencing data. So also chip sequencing data, we've got quite a lot of them already in the platform. So spread over the different genome versions, we have more than 4,000 in the public part of R2, which allows you to use any of those to really look into detail at around a particular gene. What is the association with the transcription factor, with histone modification? All these kind of things are possible, and you can visually show that in many different ways. What is I think very interesting if you start working with the chip sequencing data in R2 is we also have a genome browser filled with a lot of additional features already, a lot of different tracks. So you can, for example, use encode identified transcription factor binding sites for different transcription factors. You can use the NIH epigenome roadmap to see how the particular section of the genome is primarily used. Is it primarily an enhancer region, or is it transcriptionally active? Are there other features in it? So all of those kind of information you can just put underneath these chip profiles, and thereby maybe identify some regions that are of interest. And next to just using the genome browser to run these types of analysis, you also can use transcription start site focused analysis. And here all the regions of the genome have kind of been aligned on the transcription start site, and then you can see, if I look at these 5,000 genes, what is kind of the pattern that is shown by this chip sequencing profile. Here one example of the transcription factor and MYC, where you can see that it really associates around the transcription start site because it's the transcription factor. And below I have a histone modification, H3K27 acetylation. So this is really something that's on histones. And a histone is typically absent around the transcription start site because it's completely filled with transcription factors. So there you actually can see an inclination in the signal. And you can also add metadata features to this. Here I've used the expression of a gene expression profile of the same cell line. And then essentially you can also see that the signal of this H3K27 acetylation is really low if the gene is not expressed, but it becomes really extensive if it's highly expressed. So also those intersections you can use, you can select and then use those kind of things to identify other parameters. And there are many different ways in which you actually can also work with the chip sequencing data. From that perspective, you can use meta-analysis on top of each other. So here I'm giving you an example of running a super enhancer analysis. So you can run a super enhancer analysis on every single sample, and then use the collection of those to find parameters that may be of interest and associated with particular regions in the genome where you could identify also interesting intersections. So I hope that I've been able to show you that you can do a lot with R2. I don't expect you to be able to replicate everything that I've told you thus far, but I hope that at least I've sparked some interest in, hey, maybe that's also something that could be in it for me. I've shown you a number of analyses now, things that you actually do by clicking and then making selections. R2 also has a whole section of something that we call data scopes. And data scopes are essentially landing pages within R2 where we've already preloaded a lot of things. So a lot of settings have been preset, and this allows you to have like a quick start. And this has been used already quite a number of times in publications where people present their data in R2 as an interactive platform where you can then play with the data that has been presented in these publications. And I'll just show you one slide on the one that we've actually recently added to the platform, which comes from the team of Connie from the pan-cancer proteome atlas. Also that data we have in the R2 platform. So this is not gene expression, this is protein data. And also there you can just simply visit this page, click on one of those tiles, and then it just goes into the analysis immediately. It has preset everything for you so that you can start exploring that data for your own research or just for your curiosity on what the expression is of a particular gene. And that way you can also explore these kinds of data. And there are quite a number of them which we are now building with a number of them with people actually in the room and in this new building where we're going to get more and more out of these data scopes out in the public domain. So, data sharing in R2. So, if you are interested and you think it's nice, you always can make use of the public information, public data, because that's just out on the internet. But it's also possible that we host data for you, and that can either be restricted to a user, so only your account can see it. It can be restricted to a group, department, consortium, or we can also publicly show your data to the rest of the world. And also in things like presets, annotations, grouping variables, these kind of things, you can either keep in your own account or you can share with other users using communities that we have in the platform. If you're interested in that, we have a whole chapter of that also in our tutorial book. And how we like to see R2 is that essentially R2 can be your kind of dataset memory. We've been doing this already ever since the beginning in the oncogenomics group, where we essentially have data coming from when I entered the group over 22 years ago already. Time flies. But essentially data that was produced there from experiments can still be used today in conjunction with the latest technologies that we have. And sometimes there are really interesting patterns that we keep on rediscovering, which normally you would forget. But since the data is there anyway, you more quickly just have a look at data from your PhD student, data from a student that has been entered to the platform. So perhaps that could also be interesting for your research groups. We already have connections also with the core facility genomics and the ACDC, where you essentially, if you run an RNA-seq experiment or if you run a single cell experiment, there's this little flag that you can say send the data to R2, and then we automatically get a copy of your data, we process it, put it in R2, and then immediately you can also analyze the data that's been generated by Daoud and his team. And then using R2, essentially everything that we are doing, we are just providing to the community. So in most cases, that means that people make free use of all the things that we are doing. But as you may appreciate, there's also a team of people, of course, behind the scenes that is actually working on this. So if you like what we're doing and you see us as a collaborator, we would really like to be involved in future grants by just having a small sum of money to keep on doing what we're doing, because many small sums of money becomes a big pile, which allows us to keep on doing what we're doing. So if you start liking what we're doing, think of us if you're writing your new grant, and if you want to get in touch, you can use R2 support to do so. And there's also a connection with the research data management that can also be of help when you're putting your project together, and also make sure that things like us, but also the ACDC team, are not being forgotten in these processes. Because typically what has happened a lot in the past is that people come to us with data that's already been created, with experiments that have been performed that are suboptimal. The money is already out because the grant was already there. And then people are stuck with a shitload of data, and we just try to make the best out of it. But perhaps it would have been better if sometimes you talk to other people that have other expertises before you start doing your experiment. And then I think that's going to be the last one. Yeah. Okay. Oh, of course, R2. I'm talking about it a lot, but there are a number of people behind the scenes that are doing a lot of work on it as well. So this is the team that we currently have, that's shown on top. And we only have been able to make what we make and to become how we have turned out to be by all the people that make use of the platform during their work and published findings citing our website. So I'd really like to thank everybody that has participated in that end. And with that, I think I've shown you what I wanted to show on R2 itself. And then the next part is, I think you're ready for a bit of coffee or a drink. And afterwards, if you haven't had enough of R2, then you're really welcome to just come back and use your laptop and see what you can actually do with R2 with your own ideas. Or just use one of the tutorials to actually start playing with it. Lieke, do you want to say something?
Yes, I can. Also for the people that now would like to leave, but they would still have some hands-on experience with the platform. We have a page that you can reach by r2platform.com/workshop. And you will find all the online educational resources there. We will also work with this page after the break. The advantage is that we will walk around so we can help you further. But you will find, for instance, online courses, as mentioned here. These links, they can be found further down at the moment. But okay, so the educational resource, you can find them there. One of the courses that is recommended that really shows a lot of these analyses that you have seen during the presentation of Jan. And then it goes step by step through these analyses, so you will perform them yourself, and that way you will more easily replicate them with your own data or other kind of data that you find publicly available in R2. So yeah, even if you can't stay, you can still visit this website and yeah, go through our educational material that you can find there. And for the people that will stay, there's drinks and some small snacks outside available. And we will, in the meantime, put some tables here and yeah, welcome you back.